Hello everyone, I welcome you back to another important lecture. And for today, we will look on toward the past paper questions that have come from this topic of the osmosis. And this is one of the fundamental mechanism that is taught to you in the chapter Movement of the Substances. What we know is that in osmosis, the water would move from an area of higher water potential to an area of lower water potential down the water potential gradient, and that would be happening across a partially permeable membrane. And this would be an entirely passive process, and no energy would be utilized in this. And if we look on toward the first question that came in the May-June of 2016, the past paper variant was 4.2, and this was question number 4 for your past paper exam. What they have to say is that there are some students and they investigated the osmosis in the raw potato sticks, and for the total of 3 marks, the examiner wanted you to define the term osmosis. So what we know is that osmosis is the net movement of the water molecule, where it moves from that area where we have higher number of the water molecule, and that is the area of the higher water potential, and they would move to an area of lower water potential where the number of the water molecules is less, and this would be done down the water potential gradient. And for sure, the water, when this moves by the osmosis, so this for sure needs to be crossing the cell membranes, and the process would be depending on the internal kinetic energy of the water molecule, and no energy would be utilized from the mitochondria. So that is why we are adding the word at the end, and the word is passively, that for osmosis no energy is utilized. Now if we were to move on toward the part B, so in the part B they say that we have got these students and they measured the mass of 4 of the potato sticks using an electronic balance. So they would have some kind of the potato sticks, and they have used this electronic balance, and by that they are basically measuring their mass. Now at the moment we don't see any potato stick being present in this dish. So the value of this apparatus, this goes as 0.00 gram, like if you were to look clearly. Now moving onward, what they say is that these students, they left each potato stick in one of the 4 different liquids for the total of 5 hours. Now if you were to look here, we have got distilled water, we have got 0.1 mole per dm cube sodium chloride solution, then we have 0.5 mole per dm cube, and at the last we have got 1.0 mole per dm cube sodium chloride solution. So for sure there would be total of 4 beakers, and in all of the beakers they are putting one of the potato sticks into each of them, and after immersing them for the total of the 5 hours, now they are again measuring their final mass again, and they are calculating that what change in the mass has occurred. So what you need to keep it in your mind is that in the distilled water, if we would see an increase in the mass of the potato, so that means that the water has entered into the potato stick. And any of the concentration which they have given us for the sodium chloride solution, we have to see it that whether we see an increase in the mass or a decrease in mass. So never you have to consider is that if you come across the word sodium chloride solution and they have given you and their concentration as 0.1 mole per dm cube in this situation, so this never suggests that the water is exiting the potato sticks. It can also enter, because the student, they usually believe that wherever there is presence of the salt, so the water would always leave the potato sticks or the potato slender, but this is not always the case. Let us move onward, and here they are asking us that we are required to be predicting that which of the liquid would cause the largest decrease in the mass of the potato sticks. So for sure we need to be highlighting, or we need to be mentioning, the sodium chloride solution, which is the most concentrated, and this is 1 mole per dm cube sodium chloride solution where the water potential is the least. So more amount of the water would escape out of the potato sticks into this solution of the sodium chloride solution, and that would account for the maximum loss in the mass. So the answer is very simple. We would say this is 1 mole per dm cube sodium chloride solution. Now if we were to move further, they say the student, they dried the potato sticks with the help of the paper towel before putting them on the electronic balance, and they are asking a suggestion that why they have done it. So when you would cut the potato sticks out of the genuine potato or the original potato, so some amount of the water is always available on their surface. So we need to be removing this excess water that is present on their surface. So after when we immerse them in the distilled water or the sodium chloride solution, then when we can get an accurate measure of how much there has been an increase or a decrease in the mass of the potato sticks. Now, moving onward, they say that after the experiment, the student noticed that the potato sticks with the lowest mass was soft and floppy. And now, when they are saying that the potato sticks have become soft and floppy and they have got the lowest mass, so this is very evident that the potato sticks they have lost the water. The water has gone out of them into the surrounding solution, and as a result of that, their large vacuole it would decrease in size, and their cytoplasm would also decrease in volume. And as a result of that, the cell membrane they would push away from the cell wall, and none of the cytoplasm of the large vacuole is now exerting pressure on their cell wall, so they don't appear turgid, but rather than they become soft and floppy. So here they have asked for the total of 3 marks that we are required to explain why the potato sticks have become soft and floppy. And the answer is very simple that the potato sticks they have lost the water by osmosis, where the water has moved from the higher water potential, which was there in the potato sticks, and into the surrounding solution where the water potential was low. And in this way, their large vacuole decreases in size or the volume, and by that, the large vacuole is no longer exerting the pressure on the surrounding cell wall, and that is why they would become flaccid or they would become soft and floppy. If we were to move further for the total of 2 marks, they are asking that the student followed the same experimental procedure, but now they did not take the fresh potato sticks, but rather than they boiled the potato sticks and now they are immersing them into the solutions. And what they measure is that there is no overall change in the mass in any of the solution, like whether they place them in distilled water or whether they place them in the other solution that was zero point 0.1, 0.5, and 1 mole per dm cube. So the boiled potato sticks, they do not show any change in the mass. So this means that once we boil the potato sticks, so no osmosis is occurring. Or other than that, we could say is that there is no net movement of the water molecule in case where they are boiled. So the answer becomes simple that when you would boil them up, so their cell membranes are damaged. And when their cell membranes are damaged, so ultimately no osmosis occur. Maybe the situation would be that the amount of water that would be leaving the boiled potato sticks, it would be equivalent to the water that is entering into their potato sticks, because we know in osmosis the cell membrane is always involved. And by boiling, when we denature or when we damage the cell membrane, so automatically osmosis would not occur. And as a result, we would not see any increase or the decrease in the mass of the potato sticks. Moving onward to the next question from osmosis. This came in the year 2020 for October, November, and the variant was 4.1. And they say water is an essential molecule of life. We are required to be completing the statement, and the marks are two. Now they are talking with respect to the movement of the water, and they say water moves into and out of cells, and we know this is always with the help of osmosis. Further, they say is that the water is known as this because it can dissolve solutes. Now we know that anything that can dissolve the solutes in it, so we would always have to be referring to water as a solvent. If we were to move to the part B, which they have shown us, they say that we have got a leaf cell and this was put into a solution. Like we have placed the sleeve cell into a solution, and the water potential of the solution was lower than the water potential of the content of the cell. Now you see that if I were to talk about the solution which was here on the outside of the plant cell and it has a lower water potential, so that would mean that the cellular content they would have a higher water potential and the water would move out of the cells, and as a result of that the large vacuole and the cytoplasm they are decreasing in volume, and the cell membrane they appear to look pinch away from the cell wall. And this is why this plant cell as of now looks flaccid or this looks floppy. Now, what they have to tell us further is that the same leaf cell, this was transferred into the pure water. And now they want us to sketch the expected appearance of the cell after it had been in the pure water for 3 hours. Now, if I just replace the surrounding solution with the pure water, so that means that on the outside the water potential would be high, and in the large vacuole or the cytoplasm of the cell, the water potential would be less. So by osmosis, the water would enter into the plant cell. And that is why the cytoplasm would increase in volume, the large vacuole would increase in volume, and the large vacuole would exert the pressure on the cell wall. So these cell wall in the sketch, we need to be showing it a little bulged out. So that means that the examiner would be convinced that the student knows that here the plant cell is becoming turgid. We are required to be increasing the size of the large vacuole in the sketch, and we would move the nucleus to little bit of the corners, and they would also be pushed these chloroplasts which they have given us. So if I were to just simply take you to the diagram for 3 marks, so you see that I have bulged the cell walls out. I have increased the volume of the large vacuole. I have increased the size or the volume of the cytoplasm as well. And I have made sure that the cell membrane, they are now in contact with their surrounding cell wall. So if you would make a diagram like this, so you would score total of 3 marks. If I were to move further, they say that we have got a plant and that plant was not watered for 1 week. And they say that this figure 1.2 that we are showing, it is a series of the photograph of the plant during the week. And you see that here on the day 0, the plant looks upright. And as the day are passing to day 3, the plant is wilting, isn't it? Now, what you are required to be understanding here is that if I would not water the plants for 1 week, so this means that the water would not enter into the cells of these plants, and when it would not enter into the cells of these plants, so this means that the large vacuole would not fill up with the water. And if this would not fill up with the water, then as a result of that, the large vacuole would not exert the pressure on the cell wall, and these plant cells they would lose their turgidity. And since a lot of leaves are being shown, so this means that here we have got the palisade cells. So when the palisade cells are no longer turgid, so they would not be able to support the upright position of this plant. Now, if we were to go to the question for the total of 3 marks, what the examiner is asking, that we have to explain how the lack of water has affected the support of the leaves of the plant, shown in figure 1.2, and we are required to use the term turgid pressure in our answer. So the answer is very simple. I have explained it above. You just need to look on toward the wording that the students they were required to write down. You would say that since the water was not provided to these plants, so it would not enter into the large vacuole. And when this is not entering into the large vacuole, so the large vacuole would not exert the turgid pressure on the cells wall of the mesophyll cells, and ultimately they would collapse. And by the end of the week, the plants they would appear wilt. Moving onward to next question that came in May June of 2023. The variant was 4.1 and the question number was 2. And here we have got the students, and these students they started to investigate the osmosis, that too in the potato plant cells. Now what he actually did, that out of the potato he has cut their cubes and then he immersed them in the water, and that water had got different concentration of the sucrose solution, and then he kept these cubes of the potato tissues for the total of 30 minutes into the water that has got different concentration of the sucrose solution in them. Now, what they actually did, that before immersing the potato cubes into the sucrose solution, they would have measured their initial mass, and after putting them into the 30 minutes, now they would have measured their final mass, and now they are calculating the percentage change in the mass, and the results are shown in table 2.1. Now, if I were to explain you the table very clearly, so in the first column, they have shown you the concentration of the sucrose. Then this was the mass of the potato cubes before immersion in terms of gram. This was their mass after immersion for 30 minutes. And here they have calculated the percentage change in mass. Now you were to see that as we move down the column, the concentration of the sucrose solution is increasing. And if we were to see the first three concentration that ranges from 0.00 to 0.40, so you see that the final mass is greater than the initial mass. So this means that at these concentration of the sucrose solution, the surrounding solution has higher water potential, and the potato cubes, they have less water potential, and the water entered into them, and that is why their mass is increasing. And we see the value for the percentage change in mass as being in positive. But if we were to look the last three concentrations ranging from 0.60 to 1.0, so the final mass of the potato cubes, this is less than their initial mass, and that would indicate that these would be the concentrated solution where the water potential was less than the potato cubes. So the water, it left the potato cubes, it came into the surrounding solution, and that is why they are losing the mass, and the percentage change in their mass is indicated with the negative sign. This negative sign would always indicate that some tissue, as in this case potato tissue, this has lost the water, and that has resulted for a decrease in mass. Now they say using the information in table 2.1, we are required to calculate the percentage change in mass at 1 mole per dm cube. So you see we are given the value here, but this percentage change in mass is not available. So how the formula would go? This is the final, this is the initial. We would subtract the initial from the final, and we would divide the value by the initial, and we are required to be multiplying it with 100 to basically see the percentage change in mass. So if I were to take you to the answer, so the final mass is 1.11 and the initial mass is 1.28, and we are taking their difference and we are dividing them by their initial value of 1.28, and then we are required to be multiplying it with 100, and the answer for us, since we have to give it up to two decimal places, so this is 13.28 that you are required to write it here. Now for the second part, we are required to use information which is given in table 2.1, and we are required to explain the difference for the results that they have given us for two concentrations. One is 0.6 mole per dm cube sucrose solution concentration, and the other one is 0.8 mole per dm cube sucrose solution. And the examiner is clearly hinting that you need to be writing the term water potential in your answer, and the total marks given are 5 here. So if I just take you back toward the readings that we have got for 0.60 mole per dm cube and 0.80 mole per dm cube. So what I see is that for this higher concentration of the sucrose solution, the percentage change in mass is far more greater for the percentage which they have given us for 0.6 mole per dm cube. Now, what you have to be basically understanding here is that if I were to have a beaker here, and if the surrounding concentration was 0.6 and that too in term of mole per dm cube, and if I were to have a second beaker, and here if I would keep the concentration of 0.8 mole per dm cube, and if I would immerse the potato cubes into both of these, so the idea is simple, that at both the concentrations, since we are seeing a loss in the mass of the potato cubes, or we are seeing a percentage change or a decrease in their percentage change mass, so this simply implies that the water has moved out of the potato cubes into the surrounding solution, because in the surrounding solution the water potential is low, and in the cubes the water potential is high. So the water would move out of the cubes into the surrounding solution, and that is the reason we see a percentage decrease in the mass. But the other fact is that there is more loss of the water at 0.8 mole per dm cube comparative to this, because at this concentration the water potential in the outside solution is far more less as compared to the water potential that we have here. And since we have far more less water molecules in this solution comparative to this, so there exists more steep gradient for the water potential. So ultimately, this is losing more water comparative to this. So this is an overall idea that we have to present in our answer for 5 marks. So how you are required to be writing the answer to such questions? You would say that 0.8 mole per dm cube solution has water potential which is far more less than 0.6 mole per dm cube. And for 0.8 mole per dm cube solution, the steepness of the water potential gradient is more, and that would cause its potato cubes to cause its water molecule to move out of the potato cubes from higher water to lower water potential. And you see, we are actually complying with what the examiner has asked us. And that is why the loss of the water out of the potato cubes or the cylinder is relatively more in this higher concentrated or 0.8 mole per dm cube solution than 0.6 mole per dm cube solution. And that is why the potato cubes, they are losing more of the mass at 0.8 mole per dm cube than 0.6 mole per dm cube. Now, moving onward for the total of 2 marks, they say that we are required to describe the expected appearance of the cell from the potato cube that has been immersed in the distilled water for the total of 30 minutes. So you see that if I would place the potato cubes in distilled water, so the water would enter into their large vacuole. So their large vacuole or the cell membrane, they would increase in size, and for sure they would put the pressure on the cell wall, and the plant cells, they would become turgid or the swollen. Like we were required to be writing the appearance, not the mechanism. Now, for 3 marks, they're asking that we have to describe how the process of active transport this differs from the process of osmosis. So we know it very well that the active transport, it uses energy which is released from the respiration, and the active transport would move the particles against the concentration gradient, and the active transport would cause the movement of the ions with the help of the carrier proteins, and none of these situations we see in case of osmosis. Moving onward to the C part, they say state the type of the plant cell that uses active transport to absorb the mineral ions from the environment. So this is fact that these are the root hair cells, because the root hair cells, they have the abundant mitochondria and they have the carrier protein. The mitochondria would release energy and the carrier proteins would allow the movement of the ions. So the answer here is root hair cell. And for the 2 marks, they ask us that we are required to explain the effect of the lack of magnesium ions on the color of the plant leaves. Now, what we need to be understanding here is that these magnesium ions, they are important for making the chloroplasts or the chlorophylls within the leaves, and those that chlorophyll pigment is green and that would make the leaves look green. So if there is deficiency or lack of magnesium, so the leaves, they would turn yellow. And why they are turning yellow? Because there was lack of the magnesium ions, and these plants, they were not able to make the chloroplast or the chlorophyll. So that is why the plant leaves, they appear yellow. Moving onward to this question, and it appeared in the May-June of 2024. The variant for your past paper was 4.3, and this was question number 1 with you. This came in year 2024. It was May, June, and the variant was 4.3. And he is asking for the total of 3 marks, that we are required to explain that how the water, it moves into and out of the animal cell. So the answer is very simple, that the water would move in and out of the animal cell with the help of the osmosis, and the water would always move from an area of higher water potential where the water molecules are more to an area of lower water potential where the water molecules are less, and that too would happen across a partially permeable membrane. If the marks were 4, so you could have added up that this is an entirely a passive process. If we move on toward the second part, what the investigators, they have done, that they have taken from the person a sample of the red blood cells, and these red blood cells, they were immersed into the test tubes that contained the blood plasma, the pure water, and a very concentrated salt solution, and below they have given you the appearance of the red blood cells, and the examiner would like you to highlight that where would the appearance of the red blood cells would be in case of these three of the solutions that we have got. Now, what we know is that the blood plasma has the same water potential as the red blood cells, so the red blood cells would not lose their shape, they would not burst, neither they gonna shrink, so we would write blood plasma right in front of it. If you were to see these cells, you see that they appear shrink. So this means that these red blood cells, they were placed in a very concentrated salt solution, and the red blood cells have lost the water as the water would move out of them into the surrounding solution. So this is a very concentrated solution, and the cells, they appear like this. And here you see the red blood cells, they have burst. So this means that here they were placed in the pure water. the water moved from the surrounding solution from the higher water potential to the lower water potential that would exist within these red blood cells, and without the cell wall, these red blood cells they are bursting. So the solution that you were required to be writing here was pure water. If I were to move further, they ask us that state the name of the molecule that lies in the red blood cells and that would combine with the oxygen gas. So you would always say this is hemoglobin. And for the last question for 1 mark, they say that figure 1.3 it shows a drying of the white blood cells, and we are required to be identifying the type of the white blood cell. Among the white blood cells, we have the phagocytes and we have the lymphocytes. But if we just try to be looking on toward the appearance of the nucleus that is given for this cell, you see this is a multiple lobed nucleus, as there are a lot of lobes of this nucleus. So this is a neutrophil. And the neutrophils, they are a type of the white blood cell that we refer to them as the phagocytes because they eat up the pathogens. This appeared in May June 2025. The variant was 4.2. And the figure 5.1 that we see here, they show you the diagram of the root hair cell. So if I were to just simply demonstrate that what lies within the diagram, here we have the cell wall and we have got the cell membranes, and here lies a cytoplasm of the root hair cell. Here comes its large vacuole. And if we were to follow the key, the cross indicates nitrate ions, and the dots that we have got, this is indicating the water molecules. So if you were to simply see, we have got higher concentration of the nitrate ions in its cytoplasm and lower concentration of the nitrate ions in the soil that is surrounding these cells. So if the ions were to move into the cytoplasm of the large vacuole, so what these root hair cells would do, that they would cause their mitochondria to release energy, and that energy would be utilized to move these nitrate ions from their lower concentration that lies in the soil to their higher concentration which lies within their large vacuole with the help of the active transport. And in doing so, these nitrate ions they are being moved against their concentration gradient, and that too with the help of the proteins that we have got in their cell membranes, and they are the carrier proteins. So this is something they are asking for the total of 4 marks, that we need to use the information in figure 5.1, and we have to explain that how the nitrate ions they are transported into the root hair cells. So we would say that the nitrate ions they are moved into the root hair cells with the help of the active transport, where the ions they are moved from the lower concentration in the soil to the higher concentration, not in the large vacuole, but rather than the examiner, they have showed you the cytoplasm. So we would say that they would move into the cytoplasm against a concentration gradient, and this process would require the energy which is released by the mitochondria through aerobic respiration, and these ions they would pass via the carrier proteins. So this way you would score a total of 4 marks. And if we were to move further, they say we have to state two ways by which the nitrogen fixation occurs. And by the nitrogen fixation, what we mean is that the atmospheric nitrogen gas, this is being converted into some ions or some form of the oxides of the nitrogen that can be used by the plants. So the two of the mechanisms involve the lightning, and the other one we have got are the nitrogen fixing bacteria that lives within the nodules, within the root nodules of the leguminous plants. For the last part, they say state the name of the process that moves water molecules into the root hair cells. So this goes with the name osmosis. I hope you would be clear with that how come the past paper questions can come from this topic of osmosis.